Hi, everyone. Welcome to the AOCS student pitch. This is for health and nutrition, one of the greatest divisions at AOCS. Really happy to have you here today. This is one of my favorite parts of the annual meeting. Just started, I think, last year, and these are really the top student presentations that we have in the competition. Typically, great research, and they're having a chance to share that with you today. Also a question and answer session. So how this works, there'll be 3 competition finalists that will present today for 5 minutes each. After the presentation, there's a 7-minute question and feedback session. So you can post questions, comments, or congratulatory notes in the chat area. And then after all the presentations have ended, we'll pause the stream for a few minutes to allow for final judge deliberations. During this time, the viewing audience will be able to vote for their favorite pitch. So, and then lastly, we announce the first and second place winners of the AOCS student e-pitch competition for Health and Nutrition Division. We want to wish the students well. Thank you all the volunteer judges and the audience for attending this event. So today we have our judges are Dr. Ran-Ressler, Dr. Hongbing, and Dr. Ignacio Vietez Osorio. So they'll be presiding as our judges. And our finalists in the competition today presenting are Snigdha Guha, Ayse, Akpinar, and Emerson Nolasco. So our first presenter will be Snigdha Guha, and the title of her talk is Dietary Glutamyl Valine in Reducing Inflammation in Endothelial Cells in a Mouse Model for Atherosclerosis. And she's from the Food Science and Technology Department at the University of Nebraska-Lincoln. And Snigdha is currently a research assistant there on a PhD track. She grew up in New Delhi, India. She completed her bachelor's and master's degree in microbiology from the University of Delhi in India. Pursued a second master's degree in food science and technology and quality assurance at the University of Reading in the United Kingdom. And currently, she's working on the role of dietary bioactive peptides in modulating metabolic diseases. Her research topic is to evaluate bean-derived bioactive peptides efficiency to reduce vascular inflammation and related pathophysiology. She is also a teaching assistant for courses in food chemistry and advanced food analysis offered at UNL. She's happy to know that you're here. So without any further ado, we'll introduce Snigdha and listen to her presentation.
Hello, everyone. My name is Snigdha, and the title of my research is Dietary Gamma-Glutamyl Valine in Reducing Inflammation in Endothelial Cells and in Mouse Model for Atherosclerosis. Now, we all know cardiovascular disease is the number one cause of death globally, and according to World Heart Federation, it claims around 18.6 million lives every year, which means 1 in every 4 deaths in the United States is because of cardiovascular disease. And it has been reported that people who do not smoke, control their BMI, eat lots of fruits and vegetables, and exercise regularly can actually reduce the risk of cardiovascular disease by 85%. But unfortunately, only 3% of people actually do all 4. So it's becoming a problem And especially the problem with the current pharmacological interventions are that they have a lot of side effects. Then people need to adhere to them for a long time. And also they're expensive. For example, vascular surgeries are very expensive procedures. Therefore, we need to look for a more preventative or alternative approach to tackle this growing epidemic so that lesser people can actually reach that extreme stage where they have to undergo such expensive procedures. For that, my research focuses on food-derived bioactive peptides, and in particular, a special kind of peptide called gamma-glutamyl peptides. These peptides are unique because they form their peptide bond from the gamma carbon of the glutamic acid instead of the usual alpha carbon. And these peptides are naturally found in foods such as beans, cheese, garlic, onion. And another interesting fact about these peptides is that they can interact with the calcium sensing receptors, which are present ubiquitously in many mammalian tissues. And this interaction has actually shown to have anti-inflammatory effects previously in gastrointestinal inflammation. Therefore, my research wants to focus whether this peptide can actually show anti-inflammatory effect for vascular inflammation as well. For that, we chose gamma-glutamyl valine as our model gamma-glutamyl peptide. And the study was divided into two parts, in vitro and in vivo. For the in vitro part, we first pre-treated the peptide at 3 doses, 0.01, 0.1, and 1 millimolar on human aortic endothelial cells, which are basically the blood vessel cells from the aorta. Then we induced inflammation using TNF-alpha. And after the treatment, we checked for the expression of inflammatory biomarkers. And we did see that the 1 millimolar dose, which was our highest dose, could effectively or significantly reduce inflammation. And this is just one of the representative graph of all the results. So this effect was also found to be dependent on the calcium sensing receptor, because when we used a CASA specific inhibitor, we saw that the previously reduced inflammation was abolished and it went back up. We also looked at some of the key molecules which were involved in the anti-inflammatory effect, which included beta-arrestin, which is closely associated with the calcium sensing receptor, as well as molecules of the NF-kappa B inflammatory pathway. Then the second part, we wanted to look whether this peptide can transport through the intestinal cells intact or not, which would be important because if we want to use it potentially as a dietary intervention. So in this case, we did see that gamma EV could pass through the intestinal cells or CACO2 cells intact with a p-ap of 1.56 into 10 to the minus 6 centimeter per second. We also looked at the route of transport, which was paracellular, that is in between the cells through tight junctions, and with a peptide transporter called peptide-1. So in vitro, it was showing anti-inflammatory effect. Coming to the in vivo part, we used a mouse model called ApoE knockout mouse, which is a classic model for atherosclerosis. So we supplemented gamma EV at 2 doses, a low dose of 50 and a high dose of 150 milligram per kg body weight in the drinking water for a total of 17 weeks. And we accelerated the process of plaque development by feeding them high-fat diet. So at the end of treatment, we did see that as compared to the positive control, there was lesser lipid accumulation in the plaques for both the low dose and high dose of the mice. And also there was reduction in inflammatory biomarkers in the aorta. So basically this suggested that this peptide was potent in showing anti-inflammatory effect. So the relevance for my research, or the long-term goal of my research, is that we could potentially use this peptide to develop nutraceutical or functional food ingredient, which would be an effective strategy in reducing vascular inflammation and thus provide a novel strategy for the prevention of cardiovascular diseases. Thank you.
Okay, Snigdha, what a great presentation and a very interesting and exciting project, inflammation is a big part of our lives, and I think the hopefully past pandemic has taught us even more about the inflammatory pathway and its influence on health. So we're going to deliberate now with the judges and take audience questions and judge questions right now. So we'll proceed with that for the next 7 minutes.
I have a quick question, if I may.
Yes, please.
Excellent presentation. I really like how you take something from food and you are trying to find a solution for basically to take the food and make food applications or ingredients in food and actually make implication in health for our disease, for example, atherosclerosis. I have a quick question. Do you have any thoughts about if the matrix of the food, you mentioned beans, you mentioned cheese, you mentioned garlic and onions. Do you think the matrix of the food will have any effect on the either bioavailability or the effect of the specific ingredient?
So it does. So basically we do eat these, like for example, we consume beans and cheese on a regular basis. But since the quantity of these peptides which are naturally present is not very much, so we do need to, so we may not see the effect which we are seeing for an isolated peptide as we are going to eat food. Because I'm also doing the same research and I'm extracting peptides from common beans. But according to my calculation, we need to be eating almost like 20 kilograms of beans to actually have this same kind of effect. That is why in my research, I'm actually extracting these peptides so we can use it as a dietary intervention. So just eating the normal food may not give the same effect as probably a nutraceutical might.
Got it. Thank you. I'm a dietitian, so I thought immediately maybe food and okay.
Mm-hmm. Yeah.
Okay, possibly I can jump in. As Snigdha, I think that's a great presentation because there's a lot of data. You showed like a cell study and animal study that need a lot of time of work. So like just following Rinat question. So like you seems like your goal is to develop this peptide into a nutraceutical. It's not really functional food. So have you ever seen any other articles or reported that the beneficial effects was actually containing these peptides? So like you said, like fermented soybeans showed this kind of effects. Like
yes.
content. Okay, but that means it might be a possibility. Just it may need more research to be here. Yeah.
So our lab actually does that. So as I mentioned, like we work, like one part of my project is extracting the peptides from beans. We're also trying to ferment the beans to see if we can increase the concentration of these peptides. Because as you mentioned, like soybeans, there is study for lactobacillus, which can actually increase gamma-glutamyl peptides in those beans. So we are trying that in our lab as well.
Yeah, you can work on that. I just like heard like you did a major like previously in microbiology, right? Into that work in the future. One more thing is like have you ever seen these reports on if this peptide can really be detected in the blood circulation?
In the blood circulation?
Yes, it can be really resolved like based on the evidence from animal study.
Yeah, they do get absorbed. So gamma-glutamyl peptides are actually present in all mammals and plants and bacteria in like basal levels. So when we we did like mass spec analysis of gamma-glutamyl peptide after like the treatment. So we did detect it in the positive control as well, but definitely like our low dose and high dose had a higher value because we were supplementing them in the drinking water. But yeah, we can definitely detect the presence of this peptide in blood serum.
Okay. Another question, like, do you think this peptide, like I saw, like you showed, like it reduces inflammation in cells. So do you think it works at this receptor level? You put like a calcium sensing receptor, or it at more downstream like signaling levels?
I think this peptide has more downstream because for the in vitro studies using the pure peptide, I did see like the calcium sensing receptor was involved, but the ones from extracted from the beans, it kind of had a more downstream effect rather than the receptor itself. But there are studies which actually shows like, I mean, there are conflicting studies that sometimes the calcium sensing receptor expression is high and the activity is higher because the activation is higher. And some reports, they do show that there is like decrease in the expression of the receptor. So I think that needs a little more research to see where exactly the point of action is.
Okay, yeah, I have thank you. I have no questions for now.
Mm-hmm. Thank you.
Well, thank you very much for your presentation. It was very clear and I like it very much. And I only have another question is if you want to develop functional food, will be the next step of your research, or what are you thinking to do in the future?
Okay, so I mean, so developing a functional food, as I said, is a long-term goal. So there is definitely many steps before. So first, once we see like this peptide is effective in reducing inflammation, we want to make sure what are the exact molecular mechanisms. So we are planning to do some transcriptomic studies because also we need to see because also drug interaction, because if people are taking other drugs and we are adding this molecule, if there is an interaction or if there is any further toxicity, although we did not detect any toxicity in vitro on cells, but we want to see like how it corresponds to like when you're actually giving in high doses to, so a lot of like research would be needed to be done before reaching that stage where we can actually use it as a nutraceutical. So I think, yeah, those would be my points.
Okay, thank you.
Thank you so much.
I have a question from the audience as well. This person would like to know, have you ever looked at synergies with this peptide and omega-3 fatty acids at reducing inflammation?
No, I have not, because I just work with the peptide, so I have not mixed like other sources. Sorry.
Sure, no problem. On digestion, do you get much hydrolysis of dipeptides, or do they pass through the gut fairly easily?
Dipeptides usually pass through easily. Also, another thing with this peptide is because of the gamma bond which is present, it is usually not cleaved, because normal peptidases usually cleave but the gamma bond, so that's why it's resistant to gastrointestinal digestion as well. And they're anyway like 2 amino acids long, so they are very short.
Yeah, they're very interesting to me. And I know even like cell culture medication production or protein production, there's a lot of use of dipeptides now in cell growth to stimulate certain proteins to be produced.
Yeah.
So, all right. I think our time's up. 7 minutes are up. And I just want to, again, thank you, Snigdha. It's really nice research. And as Ignacio said, presented very clearly. It's always exciting to learn about compounds in nature that have potentially a profound impact on our health and well-being, and reinforces why we should eat fruits and vegetables and all things awesome. So contain these nutrients. But,
thank you, judges, as well, for your questions.
All right, we're going to move on to our next speaker, and our next speaker is Ayse Akpinar, and Ayse is from Ankara University in Turkey, and then her co-authors are also from University of Leeds, United Kingdom, and Hatip University in Turkey. The title of Ayse's talk is Effect of Food Emulsions on Cytotoxicity of 3-chloropropane 1,2-diol esters. So I'd like to introduce Ayse to you and listen to her presentation.
Thank you so much. Hello, my name is Ayse and I am a master's student. I will present our research titled Effect of Food Emulsion on the Toxicity of 3-chloropropane 1,2-diol esters. 3-chloropropane 1,2-diol, that is 3-MCPD esters, are food processing contaminants and are known to be potentially carcinogenic. The main purpose of this study was to determine whether the emulsion technique increase the absorption of this contaminant and to develop strategies to reduce their absorption. First of all, 3-MCPD ester contains inedible oils such as hazelnut, walnut, sunflower, soybean, corn, and olive oil were determined. You find olive oil contains the highest 3-MCPD ester level. Then find medium coarse emulsion were prepared with whey protein isolate and 10% oil using microfluidizer. The fatty acid release was investigated using an in vitro digestion model, and the bioaccessibility was calculated. MTT cell viability method was used to perform a toxicity test. The zeta potential and droplet size of the samples were measured after each digestion phase using light scattering methods. If you look at the results, the emulsion with the smallest particle size, that is fine emulsion with 389 nanometer, result in highest rise of free fatty acid, 85% during small intestinal phase. Fine emulsion also resulted in the highest 3-MCPD ester bioaccessibility, 95%. As you can see in the chart, during in vitro digestion, the droplets increased at stomach phase for all emulsion types. When an indigestible oil such as lemon oil was added to the fine emulsion up to 30%, the release of free fatty acid decreased by up to 30% as expected. However, bioaccessibility decreased by up to 45%. The missile phases of the fine and medium emulsion had toxic effect on the fibroblast cell line, and the missile phase of the medium emulsion had a toxic effect on the various cell line. The percent viability increased with an increase in particle size and with added lemon oil. As a result, there are two ways to reduce the absorption of 3-MCPD ester in food emulsion: producing emulsions with a larger particle size or adding indigestible oil to the oil phase. Thank you for listening.
Thanks, Ayse. Great presentation. Certainly a very hot topic in nutritional oils these days, and really interesting with the emulsified size affecting toxicity. So some solid potential lessons for food production. So now we're going to turn it over to our judges if they have any questions to ask. And also, if you're in the audience, you can post your question in the chat as well.
Okay. Possibly I can go first. I just got one question here. So I saw like on your slides, like you added some lemon oil. So like so the content is pretty high. It's like 10%, 20%, or so 30%. So are there any potential issues if you add lemon oil to that much? Since lemon oil, I think it is an indigestible oil. Are there any potential issues if you add that much?
Because lemon oil doesn't contain triacylglycerol, inhibits the digestion of triacylglycerol. Therefore, the more lemon oil we add, the lower the free fatty acid release and bioaccessibility. So we use lemon oil.
Okay. Are there any like side effects or negative effects if I'm using an indigestible oil?
No. We try orange oil, but lemon oil is lower the release free fatty acid. So we use lemon oil.
Okay. Okay. Thank you.
Thank you so much.
I have a quick question. So, and maybe you said it and I missed, but will there be any effect on digestion of the oil, of the edible oil, if you add indigestible oil? And
I'm sorry. Can you please repeat question?
So I'm assuming that we will use this technique in order to decrease the risk for 3-MCPDs in edible oil. That could be perhaps one benefit that I can see. Is that a correct assumption?
These studies are in vitro condition, so we didn't use animal, not in vivo study.
And are you planning to do so, or this is out of scope?
Maybe.
Okay, thank you for your presentation. It was very interesting. And I have one question. You use lemon oil to decrease the bioaccessibility of MCPD. In what kind of food this strategy could be used?
For example, actually, refined oil use everything. So, for example, mayonnaise.
Yes, because it will be a food that the flavor of the lemon oil, it will be appreciated by the consumers.
Yeah.
Okay.
Thank you.
Possibly I jump back. Yeah, so because I saw like a very high concentration of lemon oil. So is that like a risk that it may take result in like say a vitamin? Like there were many lipid soluble vitamins like vitamin A, vitamin D. So because there were high concentration of indigestible oil may take these lipid soluble vitamins. Is that like will that cause like vitamin deficiency? Do you think?
I will try to answer you correctly.
Okay.
When an undigestible oil, we use lemon oil, was added to up to 30%, but bioaccessibility decreased by up to 45%. So it increased more than we use. So it's important.
Okay. I have no questions, Doug. Thank you.
Thank you so much.
Okay, there's no questions from the audience. I was just wondering, do you know, like in making mayonnaise, can manufacturers control particle size, or is it just kind of a random mixture when they process mayonnaise?
I can't understand. Could you please repeat again?
Sure. Just wondering if the emulsion size can be affected during food production or selected to potentially follow up on your research.
With the reduction of particle size, the amount free fatty acids taken per unit time, the small intestinal phase increase.
Okay, yeah, thank you. Well, thank you for a great presentation, and we really appreciate it. Exciting research, and we're going to move on to our next presenter. So thank you, Ayse, very much. Well, we're back. Here's our third presenter for the day, Emerson Nolasco. And the title of Emerson's talk is In Vitro Bioaccessibility and Antioxidant Activity of Commercial Standard and Enriched Whole Egg Compounds Modulated by Production and Processing Practices. Emerson is at the Food Science and Technology Group, University of Nebraska-Lincoln, and his advisor is Kaustav Majumder. Emerson gave us a biography. He's a third-year PhD student, and he's originally from Honduras. He obtained his bachelor's degree in food science and technology at the Pan American Agricultural School, Zamorano in Honduras. He worked for 2 years as a production supervisor in a palm oil refinery in Honduras. He posteriorly graduated with a master's degree in food science and technology at UNL in December 2019, working on the anti-hypertensive activity of egg white hydrolysate and its effect on gut microbiome. He's been a recipient of awards in the biotechnology and poultry research area, and gave poster presentations at IFT and the Poultry Science Association. So without further ado, we'd like to introduce Emerson.
Hello, everyone. Thank you for your kind introduction. Today, I want to start my presentation with 2 quick questions. Have you bought enriched eggs? And if so, have you wondered if you obtained their health benefits? The egg is an excellent vehicle for dietary compounds, such as carotenoids and polyunsaturated fatty acids. These compounds are incorporated into the egg through the hen's diet. Once the compounds are in the egg, they have shown to be more bioaccessible than the same compounds compared to plant sources. Furthermore, in the gastrointestinal digestion, the egg proteins are broken down into peptides, which have also shown health benefits. For example, we have the carotenoids, which have shown to reduce macular degeneration in which the retina is damaged. And this is a condition that will affect 3.5 million people by 2030, according to the NIH. Also, the polyunsaturated fatty acids, specifically the omega-3 and the peptides, have also shown to reduce the risk factors of cardiovascular diseases, such as inflammation and high blood pressure. So it seems the egg is a right vehicle to address the risk factors of both diseases, right? Previous research has focused on how 1 or 2 factors affect the storage stability, the bioaccessibility, and the antioxidant activity of the egg compounds. Here, they have analyzed how the hen breed addressed in this presentation as the shell color, the diet, and cooking methods can affect all these 3 parameters. However, there is not much information about how the combination of these factors, along with the gastrointestinal digestion, can affect the bioaccessibility and their antioxidant activity. Do we get to absorb these components when we consume them? Is it worth enriching or consuming enriched eggs? To answer these questions, we took a holistic approach in which we combine all the factors together and analyze them through a multivariate analysis of variance. And we hypothesized that the combined effects of these factors will affect the bioaccessibility and antioxidant activity of the compounds. To test our hypothesis, we first took white shell and brown shell eggs, standard and enriched, from supermarkets in Lincoln, Nebraska, within 10 to 30 days of their Julian date. We then fried them and boiled them and subjected them to an in vitro gastrointestinal digestion, in which we used pepsin for the gastric phase at pH 3 for 2 hours and pancreatin at pH 7 for 2 hours for the intestinal phase. The hydrolysates were freeze-dried and used for further analysis. Carotenoids were analyzed through HPLC using internal standards, and here we observed that white shell fried eggs had higher lutein content compared to their standard counterparts before and after digestion, with no zeaxanthin enrichment for any eggs. We also analyzed the polyunsaturated fatty acids, specifically omega-3, and here we observed that white shell, raw, and boiled eggs had a higher content of linolenic acid compared to their standard counterparts, with no enrichment for docosahexaenoic fatty acids. Peptides were analyzed with a commercial kit, and we observed that brown and rich eggs, when boiled, had a higher peptide content than their fried counterpart. Finally, we analyzed antioxidant activity using the ORAC method, and even though we didn't see any significant difference between all the treatment in their antioxidant capacity was similar to those of blueberries, famous because of their antioxidant capacity due to their flavonoids. Now, using a more physiologically relevant model in which we use gastrointestinal epithelial cells and we analyze the intracellular reactive oxygen species using only the soluble compounds of the hydrolysates, such as the peptides, we observed that the hydrolysate did not inhibit the reactive oxygen species, meaning that they were not readily absorbed or that they lacked antioxidant capacity in this model. Thus, the importance of using physiologically relevant models to address the antioxidant activity and bioactivity of food components. Finally, our study will help the egg industry in optimizing enriched egg production practices to deliver bioactive compounds and educate consumers to make informed decisions about how to prepare their eggs to maximize their health benefits. Thank you very much for your attention.
Thanks, Emerson. Great research, really nice presentation, very clear and concise. I love eggs. They're one of the most amazing foods out there. They're inexpensive, and chickens are incredibly efficient at producing them. So wonderful talk, and we're going to open up for questions now from our judges and also from our audience. So, judges, any questions?
Hi, Emerson. I will start. Very good presentation. I like it so much. And I have two quick questions. Did you know which were the diets applied to enrich the eggs?
So we don't know what the diets are because we're just taken from commercial eggs found in supermarkets. But we have some ideas about what the composition might be. For example, it has been shown that eggs that have more linolenic acid are because the hens were fed with plant-based, with a source of omega-3 that is based on a plant such as flaxseed. So in this case, we think that they are giving flaxseed to enrich these eggs with omega-3. Similarly, when they feed marigold flowers, for example, extracts to the hens, it seems that they have more lutein, but not a enrichment in zeaxanthin. So that gives us some ideas of what the industry is using right now to enrich these eggs as well.
Okay, thank you. And the other question is, did you analyze the content of cholesterol that is sometimes a concern for the consumers?
We did not analyze the content of cholesterols, and but that would be an interesting point to see if the enrichment of these compounds might decrease or increase in many cases the cholesterol content, as you say. Yeah, it is a point of concern is specifically if the consumer has the tendency of accumulate cholesterol in their body as well.
Okay, thank you.
Thank you.
I have a naive question, really naive. Why did you collect the shells specifically? Why did you analyze the shells?
So as I was mentioning, the shell is just an indicator of the hen breed that is used. Some breeds give brown shell eggs, others give white shell eggs. And we wanted to see if this had any effect on the enrichment efficiency of these compounds, and if that could affect as well which compounds were more bioaccessible than others. So we didn't see such big effects as well from the brown shell. Mostly it was from the diet or from the cooking methods.
And basically, I will just ask another question. And basically what I'm learning from your research is that for some compounds, frying is better, for example, for lutein, but for example, for the omega-3s and others, it may be better to boil them. So it's basically, we need to, again, eat in a diverse cooking ways. Would you agree with that?
Yeah, exactly. And definitely, there is not like a magic pill in which we get all the compounds from the egg. You know, it depends on what health benefits do you want to get from it. Do you want to get antioxidant activity? Do you want to get more carotenoids for macular degeneration? Or do you want to get more antihypertensive activity? It has been shown that boiling eggs is better for antihypertensive activity. So definitely it's important to keep a balanced diet and a varied diet as well.
Okay. I have one last question and then I let Hongbing to ask, I'm sorry. So what will be your recommendation for, let's say I'm a farmer and I want to have to enrich the eggs. What would be your recommendations then?
It depends. What do you want to enrich them, right? If you want, for example, more, let's say omega-3 fatty acids, specifically docosahexaenoic fatty acid, because it's not very available in a lot of diets, I would say you could use fish oil, but sometimes fish oil gives like a oily flavor. So they could use an antioxidant to prevent the degradation of the linoleic acid during storage. Also, I would recommend the farmers to make sure their eggs are handled in a proper way using the cold chain, because it has been shown as well that these compounds degrade over time, even at refrigeration. So if the supermarkets are not handling properly, the eggs will not deliver these compounds to the consumer.
Thank you.
Thank you.
Hi, Emerson. That is a very interesting talk. I just got like two quick questions. So like one, so you showed that different cooking methods, it affects peptide content. So that which means that the cooking method actually affects the digestion of the egg proteins. So what leads to this difference between two different cooking methods?
Yes, most of the difference that we observed was from brown shell enriched eggs. There are other factors that could affect as well the peptide generation. For example, if eggs are stored too long, there could be some endogenous peptides being generated during storage. So that could affect the peptide content. Or it has been shown as well in previous research that frying eggs, in some cases, can give a higher peptide content, for example, for white shell. But in our case, we observed that brown shells, when boiling, gave this higher peptide content. So those are the questions we want to answer, like if there is any effect of the hand breed on how the peptides are generated on the different production methods.
Is that possible? Like that because of the different heating the temperature during cooking? I mean, sometimes like possibly for the frying is maybe the center the temperature is even higher because that can possibly denature more proteins before digestion. Do you think that might be a possible reason?
Yeah, it could be a possible reason as well. It's important as well how long do we heat treat it, because sometimes the proteins might not, like the active sites of the enzyme might not be so exposed or readily exposed for the enzymes to cleave those proteins. So I think that is one reason as well why frying in these cases, in the case of white shell,
is that my internet?
sorry.
I lost some connection. Is that me or possibly you?
I think it's me.
Okay, okay.
Yeah, I'm
No,
sorry.
it's okay. So like another question is like, say, so what is the richest fatty acid in that?
It's usually oleic and in this case, linolenic acid when they enrich them with omega-3 fatty acids.
Okay,
So those are two of the most common fatty acids.
yeah, you said oleic acid. So like the different acts like or cooking method affects the release of oleic acid.
We didn't focus in this research on the oleic acids, as our main interests are those fatty acids that can give health benefits such as the omega-3. But there is plenty of research as well on the oleic acid and saturated fatty acids as well.
Okay, that's our 7 minutes. Thank you, Emerson. Stimulated a lot of interesting questions and ideas. And it's approaching lunch in the Midwest, so maybe it's time for eggs for dinner. So very nice presentation. Really appreciate it.
Thank you. Thank you.
So I want to thank everybody for presenting today. Again, amazing quality presentations for your student research. I know mine wouldn't have come close to yours today. So it's nice to see such brilliant young minds working hard and doing wonderful things with their professors and mentors. So now we're going to launch our audience poll, and the audience vote occurs only on the AOCS Annual Meeting website. Instructions on how to vote are posted in the chat. The audience vote is important, and it does impact the placement of our top finishers. So we encourage all of you to vote for your favorite pitch today. So, and then the scores will be tallied, and we'll be back in a few minutes to announce the winner of the AOCS Health and Nutrition Student ePoster pitch. everybody. We have results from the pitch, and our first place winner is Snigdha Guha. Congratulations, Snigdha. Snigdha.
Thank you so much.
Fantastic. Yay. Second place is Emerson Nolasco. Congratulations, Emerson.
Thank you.
As we said off camera, great job. Everybody looks like they're great at presenting, and so it's really nice to see that. So appreciate your involvement in AOCS. It's a great home for your career. It was for me, and I really want to thank you all for being part of that. So so that is, let's just check here. So thank everybody for competing. So thank our judges for their time and expertise. And the AOCS Foundation actually funded the competition, so it's a great group there. And thank you to the audience who voted for your favorite pitch. So so thanks very much, and hope to see you again on a e-pitch or at the annual meeting or in some part of AOCS in the future. So have a wonderful day.
Thank you.
Thank you. Bye-bye.